Hello to everyone, I am Cristina. I am a PhD student, and today I want to present the effect of tDCS on AG profile during a semantic motor task. We can start with the relation between action and language systems, and the link was confirmed by the cognition of language. In fact, if you sing a verb with an action content, may modulate the motor area. And in the same way, the perception of action sequencing is similar to the process of semantic information in language. In particular, in our study, we consider the event-related potential that we can call ERPs, and for Andrew-like effect that we can see on AG profile, and is produced when you see an action. And this effect is similar in nature to N4, which is generated evoked by linguistic stimuli. And in this part, you can see this is about language stimuli, and it's similar to the semantic action stimuli. And in the experiment, we use a transcranial current simulation, and we analyze the effect of this simulation on ERP's profile, and we comparing the AG profiles before and after simulation or sham treatment. 30 undergraduate students took part in the experiment, and there is 18 women and 12 men. And this is the stimuli. There is 3 sets of 16 action sequencing, and the subjects see all stimuli. And in the first part, you can see that there is the first 3 frames, it's the same, but in the last frame, in the first part, there is a congruent final frame with the 3 before. In the other incongruous final frame, the girl uses an object in a not correct way. This is another example. This is not correct. She put the water in the dish and not in the glass, and this is also not correct. The experiment is in 2 phases. In the phase 1, we have the AG registration, and the participants were required to press a left or right button of the mouse, and depending if it's the right or left, it's a correct or incorrect ending scene. This is the AG recorder. We use a 64-channel DC amplifier, and the acquisition was with the software Neuroscan. This is the channel that we use, and this is an example of the couple. In the phase 2, we use the tDCS, and the half of the subject use the tDCS simulation, the other half the sham simulation, and we use a current of 2 milliampere and for 50 minutes, and we position the cathode in the dorsolateral prefrontal cortex above F3, and the anode above the occipital area, and the side we choose on the basis of the result that we obtain in the phase 1. After the tDCS simulation, subjects were submitted to the same experimental that take of phases 1, and the AG was acquisition in the same of the phases 1. In the first part, the result is that there is a more fronto-central N4 peak, and we can see more in incongruent condition than congruent condition. And second, to estimate the localization of source, we make a morphological analysis of ERPs, and as we can see here, there is a more activation of the frontal area when there is an incongruous ending final in the action. We make an analysis of the result between N4 when there is a stimulation and when there is not a stimulation, the sham and in the phases 1. And we can see that when there is a stimulation in the frontal area, the N4 effect is less and is reduced. And here we can see in the phases 1 there is a more frontal N4 height peak, and in the phases 2 it's more the frontal but near the other, but not like the phases 1. And we can see that all areas are involved in the process. And so we can see that frontal site was responsibility for incongruent processing as relevant by the presence of more anterior distribution of N4 effect. Partial inhibition of the frontal areal reduced the standard semantic incongruent effect, and this indicated that the decrease of N4 it's important indicator that because we can see the relevant role of the prefrontal area in this semantic process. And in the phases 1, the phenomenon is more than in the phases 2, and we can see that in the phases 2, all the area are involved in support of the frontal zone, and it's like a brain plasticity, it's like a support of the other area. And we can see also that the neurostimulation influences the electrophysiological measure, and the direct current stimulation have some effect on brain function. And this measure may be basic for the comprehension of the characteristic and the potential of the tDCS, like a brain non-invasive stimulation. This is some references.